Okay guys, we're going to have a quick look at mutations today, and as you guys know, mutations don't really work all the time. We have these images like of this snake here where some fantastic examples happen, where these freakish things happen, but we know 99.9% of the time when you change the DNA code it just doesn't work anymore. What I'm going to do today is I'm going to run through some of the vocab quite quickly. If you want some more detail about the vocab, perhaps have a look at some of the other videos and some of the text. This is really just a brief summary after you've explored those things. So first off, mutations can be induced or spontaneous, and induced one is the one that's caused by those UV rays or radiations or toxins or chemicals, something that some ways encourage that change in the molecules so they don't fit together the same. And spontaneous is probably the same process but happens naturally, and we all have mutations in our bodies. A lot of them don't have any effect, and a few of them have some minor effects. And now they can occur in two places, germline or somatic, and I might deal with somatic first. That's just in the normal body cells. And if a mutation occurs there, it doesn't have really much effect apart from that cell and the cells around it. When it occurs in the gamete, it affects every cell of the new organism. So it has a big effect. And we can imagine, like in the picture here, if a sperm or egg have a mutation, that's going to affect the genetic makeup of the zygote in that first cell of the organism. So let's now have a look at types of mutations, and there's a couple here. Polyploidy, which we might be familiar with, our example being triploidy, which is 3N. So polyploidy is when we have multiple sets of the chromosomes, so 3 or 4. Some plants can survive in those conditions, but usually in animals it means the organism's pretty dysfunctional. Next off we have aneuploidy, which is a multiple set of one particular chromosome. An example being Down syndrome, or having 3 sets of that number 21 chromosome. And there's various syndromes that are associated with having multiple numbers of a particular chromosome. The next two we're going to look at in a bit more detail, which is block mutations and point mutations, which there's a few varieties of each. And firstly, a block mutation. Now, a block mutation is really a section of chromosome, or a chunk of chromosome, a section of chromosome that can be inserted, it can be put in, deleted, duplicated, or translocated. So let's have a look at these. So duplicated, firstly, is when we repeat the same section, so it repeats twice, and we can see from this banding pattern it repeats twice. And inversion means we sort of flip it around, so this end here becomes this end. A deletion's when it's completely removed, and that's probably going to have more significant impact than the other two that we've just talked about. An insertion is where we're inserting the section into another chromosome, and that's and here we're referring to that translocation as well, where that section of a chromosome is moved to another section, another chromosome. So say from the number 7 chromosome to the number 13 chromosome. So it's allowing it to function in a different way. So now let's move on to point mutations, and we can use some similar terms with point mutations. These are the actual nucleotides being, rather than large chunks, we're talking about a nucleotide substitution, deletion, or insertion. And let's have a look at these in a bit more detail and the impacts that they might have. We have the original DNA strand across the top here. The one where the mutation has occurred across the bottom, we can see this highlighted nucleotide here. In this case, we have this C being replaced by the T, the cytosine being replaced by the thymine. Now, if we read this original strand in its triplet codes, we could read it off in its threes like this. And we can imagine each one of those codons would code for a particular amino acid. When we look on the bottom, what we're going to notice is the first 2 nucleotides, no problem at all. Where we start seeing some difference is in this 3rd nucleotide here. And we're probably going to have this produce a different amino acid. We're going to read TTT instead of CTT. Now, that would be the case for a substitution. Now, let's take a couple steps back and imagine if instead of a substitution we had a deletion. what we'd have here is this triplet code being read TTC instead of CTT. The next one, instead of CTC, we read TCC and CAA, AAG, and so on. So you can imagine that everything from this point onwards down the DNA strand is going to be read wrong, and we call that a frame shift. Now that can occur from the addition or deletion of one single nucleotide. So here we have a couple of descriptions of the outcomes of point mutations. They can be silent, nonsense, or minsense. Now let's have a look at silent first. Now silent might be a change, and we can see we've changed from TTC to TTT. However, because some codons code for the same amino acids, sometimes it might have no effect, or the effect might be very minimal, and we might not change the shape of the protein, and we call that a silent mutation. A nonsense mutation is when that new code changes from TTC to ATC, causes it to be a stop codon, and that just stops it being read. There's no sense, it's not being read. And a missense is when it is being read in a different code. It was TTC, now it's read as TCC or TGC, and they code for different amino acids, changing the sequence that we see. Okay, good luck with studying mutations. I really suggest you wrap your head around the vocab. There's quite a few things that you need to learn. You need to speak the language of mutation, and once you get that, I think the concepts are pretty easy to grab. Okay, good luck.